This is Unit 5 Biology revision video, which is covering everything to do with digestion. So this is a standard picture of the human digestive system, and we can see that it's clearly labelled. I'm going to go through this and go through the process of digestion whilst looking at the function and the different types of digestion that happen throughout the digestive system. So first of all, we start in the mouth. Here we've got salivary glands as well as teeth that will help to break down the food that we eat. Now the whole point of this is that the food that we eat is broken down into smaller molecules from the large ones that they are in the food, so that they are able to be absorbed into the body and used to build other molecules that we need to build in our tissues, and also for the process of respiration. Now in the mouth, the teeth will mechanically break down the food. This is called physical digestion. So chewing the food will make it easier to swallow. However, some food that we eat can be quite dry, and that would make it difficult for it to make its way down the esophagus. So we have the salivary glands here. Now they secrete saliva into the mouth, and the saliva contains enzymes, and the enzymes will start to begin to digest chemically the food that is in your mouth, as well as providing moisture to allow it to go down the esophagus easier. So after being swallowed, the food will make its way down the esophagus. Now this happens as the esophagus is essentially a tube of muscle, and the esophagus will contract and relax and squeeze the food down the esophagus to enable it to get to the stomach. And this process is called peristalsis. Now when we make it down to the stomach contains stomach acid, which will start to chemically break down the food, and also it contains enzymes which will also chemically break down the food. The stomach also undergoes a churning action, which will mix the food, break it up even further, which is a form of physical digestion. At this point, the stomach has broken down the food further, has digested it further, and what we're left with is a substance called chyme. And this is a liquid substance that will go through to the small intestine and will have the nutrients absorbed out of it. Now that's not the end of it when it enters the small intestine, because the small intestine also contains further enzymes that will break down the food even further, which is chemical digestion. And also the liver produces bile, which is stored in the gallbladder. Now bile will enter the small intestine, and that has its own function which we'll talk about later. Now as the food works its way along the small intestine and the nutrients are absorbed into the bloodstream, it finally enters the large intestine, where the last part of digestion will happen. Now what happens here is that the digested food is still quite full of water, and the large intestine will absorb the water into the bloodstream, and then you would be left with solid faeces. That would enter the rectum where it's stored, and when needed it is released via the anus. And this process, due to the fact that none of the waste has been made inside of your body, is called digestion. Now as mentioned in the previous slide, chemical digestion happens throughout the digestive system. This includes where carbohydrates, fats and proteins are digested by enzymes. So this is what we're going to look at next, the specifics of chemical digestion, the enzymes involved, etc, etc. So the three different types of food that you need to know about when it comes to enzymes and where the enzymes are found are carbohydrates, fats or lipids, and proteins. Now you can tell when a word is an enzyme because they end in ases or "ase" if we're just talking singular. So carbohydrates are broken down, are chemically digested by carbohydrases. Fats or lipids are broken down or digested by lipases, and proteins by proteases. Now these two will be quite easy to remember because they're very similar to carbohydrates and proteins. However, to be able to remember this, you will need to remember that fats are lipids. So, where do they work in the digestive system? Well, carbohydrases will break down carbohydrates in the mouth, so in saliva, and in the small intestine after being released from the stomach. Lipases work entirely in the small intestine, and they will work alongside with the action of bile, which we'll talk about shortly. And proteases are found both in the stomach, along with the stomach acid, and also in the small intestine. So as you can see, all three are found in the small intestine. Carbohydrates is only in the mouth, which you can sometimes taste when you're chewing bread. It can sometimes taste sweet if you chew it for so long. And only proteases are found in the stomach. Now what these do, the products of them chemically breaking down these food molecules are as follows. So carbohydrates, when they break down carbohydrates such as starch, it converts them to a maltose sugar, and then further into glucose, which is a single simple sugar. Lipases break down fats into fatty acids and glycerol, and proteins break them down into amino acids. Most of these will then go on to build more molecules, further molecules in the body when they're absorbed into the bloodstream. Things like glucose will be used in respiration. We know that pH will affect how enzymes work and can, if too high or too low compared to the optimum pH of that enzyme, cause it to become denatured. Now this is especially true when it comes to proteases, where they're found, for example, in the stomach. Now in the stomach, the pH is between 1 and 2, and that's extremely acidic for enzymes, so its optimum pH will be around this number. However, the protease enzyme that works in the small intestine will have a pH value of around 7 to 8, a neutral value. If you take the enzyme from the small intestine and tried to put it into the stomach as a protease, then it's obviously going to become denatured because it's outside of its optimum. So pH values do affect, even in the digestive system, how these enzymes will work. So as I said earlier that the liver makes bile and it's stored in the gallbladder, we're going to look at what bile does now. So as you may recall right from Chemistry Unit 1 module, is that oil and water, fats and water do not mix. They will separate out. However, fats need to be digested in the digestive system. But what we'll get is rather large droplets of fat. Now, bile is secreted from the gallbladder into the small intestine, and it will mix with the fat and will bind them like an emulsifier. Now what this will do is create smaller droplets of fat. So if these are our smaller droplets as a result of the action of bile, compare them to the larger droplets. They have a larger surface area now on the fat for lipases to come along and break down the fats into fatty acids and glycerol. So that is the function of bile within the digestive system. And the last thing that we need to look at is the adaptations of the small intestine when it comes to being able to absorb nutrients from the faeces into the blood. Now, the first one is that the small intestines are really, really long. So there's plenty of time, plenty of surface area for absorption to happen across. In this picture we can see that we have these structures that are like folds in the intestine, the small intestine. These structures are called villi, and it is folded intestines, and this increases the surface area, so there's a larger area for absorption again into the bloodstream of nutrients. Now further to this, we also have on the villi further protrusions called microvilli. Now I'm just drawing this here just as an example to show you, but what you can see is that these villi are finger-like projections coming from the small intestine, and then they have the same structures along the villi. So this further increases the surface area for absorption of nutrients into the bloodstream. And the last part, the last adaptation that the small intestine has got is that it has a really good blood supply. There's no point having such a large surface area for maximum absorption of nutrients if you're not going to have a good blood supply. So this will really help with getting as much as possible out of the digested food.